Hello everyone. So today I'm going to be guiding you through the manual human peptide inspection and verification process within our SpotMap MS software. So the approach we've taken with our SpotMap MS software is we have a very high false discovery rate. So the problem we're trying to solve here is obviously host cell proteins can be present in your sample at vanishingly small amounts compared to your drug substance or other substances, yet still at those very small amounts they can have a very large impact. You can have 1 part per million of a host cell protein causing serious immunogenic or stability effects within your drug substance product. So we're really looking for very, very small signals and trying to be really very sensitive about this because the things that we're trying to detect can potentially be very dangerous. So we do this by setting the false discovery rate very high so that even if there's even a slight chance of that host cell protein being present in your sample, we include it. We then present the user the tools to very quickly validate and confirm or deny the presence of those host cell proteins within the sample to make it as easy as possible to get rid of those false positives. So we always err on the side of caution. We always aim to include something that may not be there rather than miss something that that is there, basically. So this big filter, this bucket where we've kind of picked up absolutely everything that might even possibly be within your sample. We then use the peptide inspect workflow and a human in the way, a human in the loop, to validate and verify those matches based on your MS1, your MS2 data, whether it's a unique match, how many unique matches, and how many traces that you've got this match within. So like I say, we've presented the data here. We've got the data on the right-hand side, which is kind of your quality metrics for your operator to go through and to make the judgment on. And the tools we've got to very quickly accept or reject that match are just, you can see at the bottom right-hand side here, our I key, our O key, and our P key. So the idea here is that you keep your operator looking at the screen, your analyst is looking at the screen, looking at the curves, the data, all the metrics they need to make a decision, and then they can scroll through just using the up and down keys and then pass or fail these peptide matches. So do they feel confident that that's the correct match? So we can pass it, and you can see as we go through on the list with the peptide matches, we get a tick or we get a cross. We can always have an unknown if we want to revisit those or we want to check with a colleague if we're not certain on the quality of the data that we're getting at that point. And as we start to build up this picture of accepted matches or rejected matches, you can see that we start to build up this column here based on the color of those. So if I add in a few more failed matches, you see we get a little bit of red in there as well. And we can do this on a per trace basis. So we can do this for every single trace that we've got. So there are some instances, and I think in other videos I've talked about this and the way we display this, where some host cell proteins might only show up in one trace out of a triplicate, so one trace out of 3. Typically, the industry would only accept there being a true positive result if it was in 2 out of 3 traces. But we have the ability to run through each individual one and assess them separately, assess them differently. We can see here that for this particular peptide match, you can see we've actually got the number here saying it's present in every single trace. We have got a quality score here. It's not a unique peptide, but we can see actually for this match in this trace, so trace number 2, we're getting unreliable data. So the data is not of a high enough quality that we're considering it reliable. So that would probably be, we haven't got enough data to make the assumption, so that would just be a straight away a fail. This one, we've got a few different quality metrics to say it's an adequate match. The quality is low and it's quite poor, but again, we present the operator with all of the data for them to make the judgment call themselves. So the blend within SpotMap is to try and do as much of the processing as automatically as possible and reduce interoperator variability, reduce the number of different settings that a user could change to try and increase or increase the idea that if you give the same data to 6 different operators, they're going to give you the exact same HCP report, the exact same HCP numbers from each experiment. So this color coding system also comes through to our report when we go to export our report. And you can see here we start to build up these colored columns, and it makes it very easy when you export your report to show this to someone. To say, look, we think we've identified these host cell proteins. It's in all three of our traces. The quant stays relatively stable amongst all of our three traces. And we've had a human look at the data quality to really confirm that it is present in all three of these traces. So we've got a number of checks and balances from a QC point of view to say the software's automatically detected it. We've had a human validate the detection and validate the data quality is high enough that we can make that assumption and say, yes, I think this is definitely present within each of our traces, or 2 out of 3, or maybe only 1 out of 3 is good enough quality. So by having this blend of automation, but also with a human in the loop, having eyes on and reviewing all the data and giving users the ability to review all that data super quickly, means we can get the best of both worlds. We can get, we're dealing with host cell proteins here. They can potentially be very dangerous to people, but they can also be very costly to businesses in terms of drug stability. We need to be certain, we need to be sure. So by having this contextual automation, these great reports, this brilliant kind of color coding system and graphing quality, we've taken something that would normally just be kind of a spreadsheet full of numbers. We've given it context, we've linked it to where we think the danger lies, and we've provided a huge number of tools to ensure a high level of quality, because things like this maybe going to the FDA or the MHRA, depending where you are in the world and what governing body you're under. By having these tools and making it super easy and kind of de-skilling it, we're really trying to hammer into the quality aspect of this work and being able to make it as approachable and easy as possible for people to do this that might not be, proteomics experts or very high level mass spec experts, but still need to get a reliable, rigorous report for host cell proteins within their samples as part of their, workflow. I hope you enjoyed this demo. It's kind of a little bit of a quicker one than usual, but I think I've given enough information for you to kind of take a trial of this software and try it out on your own data. And please feel free, if you have any questions, to drop them in the comments below. Reach out to us directly. If you do want a free trial of the SpotMap MS software, please check out the link in the description below and reach out to us, and we can give you a free trial and kind of guide you through the software, the ethos behind it, and how much it can reduce your current workflow, and how quickly it can get you from sample to curated host cell protein analysis report.